Okay, breakout room 1. First, we're breaking the rules. We've got more than one slide because we didn't know this would still be up here, so we thought we'd entertain you. So what's missing from the diagram that we were given? Number 1 was time. Number 2, I just added this morning because it doesn't actually have space. It just had interdisciplines. It was missing the human discipline, which is at the bottom of our list. It was missing the connections to terrestrial, coastal, benthic surface, and ice processes. So the boundaries. And it had no arrow between physics and biology, and it had no arrows that went all the way to the bottom. So that was what was generically missing. Okay. Then we got into that. So let's see if we can summarize. So these are the 2 diagrams that are on the board. What pathways do we know well? After some discussion, we decided the answer was none. Cut off the discussion. What pathways do we not know well? All of them. So there's an edge to our knowledge in everything. So the horizontal structures, this is most a depth perspective versus an isopycnal perspective, where things are flat versus tilted, along isopycnals, across depths. Microbes, fish, benthic fluxes, topographic influences, mixing processes, the eddies, the mesoscale, and the submesoscale structures. I think those come in from every discipline. Things like hotspots, human activities, Lagrangian versus Eulerian perspectives don't show on the diagram. Fluxes versus standing stock for the export, and energy fluxes, and everything else on that diagram. How much do we know about exchanges across disciplines? I added a little bit to this morning. We said there are many gaps, but I'd say that my main takeaway from just listening to the whole discussion is that the biology sector of the room is thinking vertically, and the physical oceanography overturning pathways group in the room is thinking on really large horizontal scales and sort of overturning to the abyss. So we get to the abyss in very different ways. The biologists by exporting things down, and the physicists by going thousands of kilometers out of your way and back. So how do we tie those 2 together? That's really that's my main takeaway on the gap that we have between the 2 disciplines. And there's certainly thinking on much smaller scales on both sides, smaller scales in physics. I'm not so sure I was hearing the big scales in biology, but maybe that's because that's not what I am. Fish were not in there, so they're big scales. I think future directions, we did get into this. We're thinking about 3-dimensional biological exports. So going into that 3rd dimension, and time, of course, makes it 4. For the larger scale group, a large scale baseline on the overturning, and particularly in the Southern Ocean, which just happened to be what we were talking about in there, and abyssal formation of bottom waters and so forth. A lot of enthusiasm for deep exports. Yes, you hear it. Here's the enthusiasm. And then I'd say bullet 4 is that tying in all corners of the room, looking at smaller scales, mesoscale, submesoscale processes. How do those impact the large scale? How do those impact the small scale? So that's me. That's group 1.
Okay, so our group had a lot of really roundabout conversations. And here are some things that we put on the summary slide. We talked about many, so if anyone else from my group would like to chime in, please do. So some of the key pathways from the diagram that we thought were missing were, again, just like the first group, time and the different spatial scales. We thought it would be cool to have different diagrams for short-term processes versus long-term processes. We also thought that the arrow to the seafloor was missing, so we added that. And bathymetry along the seafloor was one important thing that we thought should be added too, and the synchronicity between these spatiotemporal scales. So we thought that there were some pathways that we knew sort of well, and most of these were on a larger scale. So the large scale physics of ventilation and circulation to the deep ocean, large scale patterns of MPP and nutrient distributions. But we really struggled to come up with some of those what do we know wells at the end when we were making this slide, because there are so many that we don't know very well. We didn't put as many as group 1, but we talked a lot about biological pump transfer efficiency and POC fluxes to the deep ocean, and how do we quantify export to the deep ocean. We talked about mesoscale ventilation and circulation. We also talked a lot about fish and, importantly, other non-copepod animals. And then, yeah, impacts of DVM, upwelling pathways. We had a lot of conversations about like model development and how the modeling world sees a lot of these missing or views a lot of these missing processes. And we said that comparing methods is very challenging, especially on regional scales. And then we did get into talking a lot about recommendations, and we thought that the biggest priority was to expand BGC Argo and Deep Argo program, and add more sensors to the floats. You know, if money wasn't a thing, we should be doing all of this. And sensors on the seafloor. And then one other important perspective that we thought will be important to maintain with the recommendations is, you know, recognizing that, you know, the sensors that you have are going to tell you one thing, but might be leaving out some other interpretations. So maintaining that variety is important. Yeah, I think that covers it. Cool.
All right, classroom 2. Come on down.
All right. Hey, everyone. A lot of what we discussed has actually already been said, particularly in terms of the pathways that we felt were missing. One of those being air-sea interactions, but also the connection from the water column into the deep sea floor. One thing I don't think we've really discussed is the potential flux from onshore and land in riverine systems that potentially could end up in the deep sea. That was missing from that diagram, but also in general, just lateral redistribution of carbon and other nutrients from that could be going on in the near surface waters as well as in the deep sea. As far as the second and third questions of what do we know well and what we don't know well, we kind of just decided that we didn't like those questions because we didn't feel like we know anything well. And we had a lot of discussion. And after that, we decided to kind of just rank things based on what we feel that we know relatively well, down to what we really just feel we don't have a grasp on at all. So we felt that we had a pretty solid grasp on deep water formation, particularly how water is going down, but not how it may be going back up. We felt we had a good grasp on the solubility pump, and then as well as anthropogenic carbon uptake into the ocean and air-sea fluxes. On the bottom, we kind of talked about how there could be a flow from what we do know well to what we don't know well. So in that bottom section that starts with physics to chem to bio, we felt that we have a better understanding of the physics and then moving into the chemistry, and then we just know less about the biology in general. And speaking in broad terms, we kind of felt that that was just the general trend. But we also could talk about this in terms of scale as well, where we know more at a coarse scale, and we should take a better look at the finer scale processes. But then talking about temporal scales as well, we probably know more from the steady state perspective, and there's a lot of episodic pathways that we may just be missing in our understanding. But then we also probably know a lot more about stocks than we do about rates. And that gets into this discussion of, do we really understand pools or do we really understand pathways? And I think what we really want to understand is those pathways. But then we also had an interesting discussion about, we've focused a lot on carbon in this meeting, but there's other nutrients as well. We brought up oxygen, nitrogen, phosphorus, alkalinity, and then we probably know a lot less about iron. I think Charles called it the Wild West. So yeah, we in general, we kind of wrapped it up by talking about how we would love to see physics and biogeochemistry and biology kind of work together and integrate using observational data as the thing that links us all together. Is that very good? Getting the thumbs up from Charles. Good.
All right, and then we have virtual breakout room number 1. Folks out in the cloud.
Hi, can you hear me okay, Leslie?
Sure can.
All right. Well, a lot of what I have on this slide has already been said as well, which is a good sign. We all agree, basically. In terms of the diagram, we felt that dividing it by discipline was maybe not as useful as dividing it by process. Instead, so our sort of baseline suggestion was starting with something similar to what Tim DeVries had in his presentation. So splitting it up into what he called the mixing pump, but we were saying mixing kind of has these other meanings in physical oceanography. So I don't know, maybe calling it the physical pump, then the migration pump and the gravitational pump. And then the big thing that we felt was missing was a continental slope and thinking about sediment transport in benthic communities. And we did kind of have the implicit assumption, I think, following on from the talks yesterday, that we were mostly talking about carbon here. And then we also discussed the arrows between the disciplines in the diagram, and you know, we wanted to clarify, you know, are we talking about how like different physical processes and biological processes, for example, interact with each other? Or are we talking about what we can learn from each other? So for example, Sarah Perkey brought up, you know, tracers sort of in the biogeochemistry sphere can teach us a lot about the physics. So that was one example of an arrow going from BGC to physics, but not necessarily a process that's interacting. Okay, and then similar to the last group, we, you know, also said we tend to know the physics a little better than the chemistry, a little better than the biology on the global scale, because of how many observations we have and sort of how easy they are to do. But maybe when we think about a meter of mud, we know more about the ecology there than really the physics that's going on. Okay, and then a couple other points that came up are that the interaction between the scales is really tough. So kind of like the other group said, you know, small scale, small time scale, sort of episodic events and heterogeneity are really important. But to get a grasp of how important they are, we then need to integrate them over the large scale. And then also following water parcels or having this 3D Lagrangian view is important. So it comes back a little bit to what Lynn was saying, you know, that it's not just up and down, it's all around, and we need to sort of bring these viewpoints together. That's all I have, unless anyone else in the online world feels I missed something.
All right, thank you. Awesome. This is all great. So we actually, it's 8:36 now, and we're not slated to start a talk until 9. I think this is probably the first time ever I've been in a meeting with breakout room recap, so we did not go heinously over. So you guys have earned yourself the opportunity to have 24 minutes to discuss this a little bit more and reflect, you know, especially cross-room pollination. Anyone, you know, after sleeping last night, have some more thoughts, or after hearing other people's, you know, discussion points in their report outs, have any additional thoughts?
Thank you. Yeah, I thought all of those were fantastic. And yeah, I really agree with most and if not all of the ideas, especially I think the Lagrangian tracking and the multi-scale are really crucial issues. And I'm not just going to point like the two places where I disagree, because I don't know what we're going to do with the slides, and maybe we need to discuss a bit more. I think the first one was the breakout one, classroom one, where we're saying we understand everything, all the except the animals and the non-copepod animals. That's very true, but I don't want this to imply that we understand copepods, because there are still many things that we don't know about copepods, and yeah, we shouldn't like mark them as like we know everything. And the second was for the classroom two. And I think maybe that's the biological oceanographer in me talking, and it's very different for the physical world, I don't know, but we said that we understand the stocks very well, but not necessarily the rates. And for the biological carbon pump, that's kind of the opposite, where we just compute export rate, but not sequestration, or very rarely.
And I don't know, Stevie, do you want to recap on the copepod? My impression was you said, we don't know any of this. We just particularly don't know the squishy bits.
Yeah, exactly. When Colleen and I were talking about this, we were saying that in general, copepods have been more of the focus than other. Yeah. Since I'm the one who said that, I think it's more like when they're like when the animals are included in a model, it's usually they kind of behave more like a copepod. So it's more from that point of view, not like, oh yeah, forget about the copepods. They're not important. Of course they're important, but you know, like models assume that all animals are behaving like a copepod, but you know, it's more from that point of view. Yeah.
Pointed out that they are easier to sample, easier to keep alive. So we know more about them in terms of the biology, the rates, how they feed and so forth, but it's clearly not everything that is down there as we saw yesterday.
Yeah, and I think to kind of put this in our workshop mantra, it would be, yes, copepods are important, and there's squishy bits that we need to look at. I should stop calling them squishy bits. There's gelatinous, amazing animals in the water column.
I guess picking up on this, it seemed like many groups said that nothing falls into the category of known. And we had a little bit of a discussion of that in our group of like, what does it mean to be known? And we said that means that you know the mechanism and you have a quantitative estimate of how important it is. And I think thinking in the sense of like, well, there are many things to know. How do we prioritize what needs to be known for a particular objective? And I think thinking in the context of this workshop, we were saying, thinking the particular objective of how does climate change connect to the deep ocean, maybe there may be some sense where you might say, well, we know this well enough, or we think we know this well enough to answer that question and maybe not answer a different question. And so, and in that sense, also not saying that the work that people are doing to answer other questions is inconsequential.
No, that's a great point. And I think this is one of those things where the scientific lexicon versus the, lexicon of the rest of human beings is a little bit different. And it's our struggle to say that we never truly know anything because we're always seeking to learn more, to refine our models on stuff. And this is why when people like in the climate change discussion, they're like, well, scientists don't know. And we're like, well, we kind of do know. We just don't want to say no. So I love the rankings of stuff we have better handles on than less handles on. And I think that that's like a really important place to start as we figure out the missing pieces and the gaps and how to extend our knowledge into the pathways. So, all right. Awesome. Good, Colleen.
Kind of on the same topic, I was thinking about how yesterday, your intro talk and also Lisa's talk talked about like, well, why do we care about this? I mean, there's a lot of scientific reasons, but there's a lot of motivations to inform deep sea fisheries or like how climate change is going to impact those fisheries or deep sea mining or MCDR or whatever. And so I was thinking about, what do we need to know? How well do we need to know things in order to inform those big picture motivations? And it might be helpful to think about it in that context.
Yeah.
Can I?
That's a great point. Yeah, Chris.
Because some of that's piggybacking on what Colleen was saying that
some of the discussion we ended up having is like, what are we even talking about here? Well, like connecting all these things. It's like, is it carbon? Because carbon came up a lot on reporting back today. And kind of the way I see it is climate, right? And carbon is just used as a currency that we can convert things. So you can go very deep into all the different biogeochemical mechanisms and like what's going to be important here and there. But if we want to look at the big picture, right? Like a lot of what's going to be missing is, nutrients. So how's, different processes are going to redistribute nutrients? How are different perturbations going to do that? But if we need a currency to put it all together and connect to the physics, I think that's where carbon comes in handy, because that's what causing climate change to begin with. Not all of it, but a lot of it.
That's great.
Just-
That's great. Yeah, my background is biogeochemistry, and my fisheries friends, I would always say, your fish are nothing but sacks of carbon. What time we got? We still have more time. Any other comments? I love that the phrasing that carbon is the currency. It's like kind of how we enumerate things. You saw from an OCB workshop? Plug for OCB. It was called Seesaw? Oh my gosh, that's amazing. Man, now deck sports is sounding less sexy. Okay. Yeah, Charles.
I'm still processing my coffee, so I'm keeping up with everybody. But it seems like one message I was hearing was that, I think when we rank these things on more known versus less known, there is a natural tendency to think about, okay, there's going to report, we're going to prioritize the less known things. And I'm not necessarily so sure that's the case, because there's known and less known. There's, do we think this is critical or do we not think it's critical? There's, do we have an opportunity to know it better with the technologies we have, and versus, well, we don't know it, but we really have no avenue to make inroads on that at the moment. So I guess I hope that as we sort of synthesize this, that we don't end up doing this laundry list of these are the things we know less, and that therefore we should prioritize them. It should be a more kind of fulsome consideration of the levels of knowing, the likely importance, and our pathways to improve that knowledge.
Yeah, well, and another important thing is that we might know something now, but we're in a perpetually changing world, acceleratingly changing world. And so I think prioritizing the things like, we may have a really pretty good understanding of overturning circulation, but that might all change. Where's Jake and his arrow disappearing? Jake ends by saying, what if an arrow just disappears? And we're all like, too late, they didn't do that. Yeah, so we need to prioritize stuff that we need to continue monitoring, continue knowing. Oh, you got two mics.
Sure. I mean, along the same lines, on one of the lists, it said, I think we know deep water formation well, or something like that was I don't know which group it was. But coming from the physical side, and especially seeing Sarah's talk yesterday, and with the OSNAP observations coming about now, I thought we knew deep water formation well. But now I'm not so sure anymore, because the locations in where they are forming now, there's still a big debate whether Labrador Sea or the eastern subpolar North Atlantic. That's still being discussed, and we are still discussing. It's a new development. And if there are really 4 deep water formation regions in the Southern Ocean, which we have been focusing on the Ross Sea and Weddell Sea, not doing anything with the other ones, and I have no idea what the models are doing. So I think, as we know more, we now know that we know less, if it makes sense. So.
Yeah, I had a similar comment to Gökhan, except I was thinking of the example of mesoscale eddies, which we know a lot about. We had like process studies on mesoscale eddies going back half a century, and yet we still can't model them very well. And that's one of the major biases in models, is that our parameterizations for mesoscale eddies aren't working. And of course, that's going to affect biases in every other tracer in a model.
No, that's great.
Okay, welcome back everyone in the room and online. So first up we have Mike Stukel, and he's going to be talking about the many faces of the biological carbon pump from FSU. Welcome, Mike.
Thanks. I'm going to do my best to convince you that I have no idea how the biological carbon pump is changing. So we've already seen this figure once, but this is from the Boyd et al. Review, where we're basically looking at the different potential pathways of the biological carbon pump. And I like to kind of think of it as we've got the biological carbon pump, or the gravitational pump, which is kind of driven by particle production and sinking speed, and the lability of sinking particles is really important. Lability really refers to basically how yummy the particles are, how quickly the bacteria chew them on. So are we talking about, M&Ms, or are we talking about Brussels sprouts? Next, we've got the eddy subduction pump and the mixed layer pump. I kind of think of these together as being driven by circulation. But again, lability is really important because it determines how long the organic matter will survive along the physical pathways. And finally, we've got the mesopelagic migrant pump and the seasonal lipid pump, which are basically the two pumps driven by the ecology of zooplankton, by the fact that they migrate up and down either daily or annually. And we can kind of put some estimates. These are the estimates that Boyd put on these in his review paper. And we see that the biological gravitational pump is kind of the most important, but the others are potentially adding up to just as significant, and those other pumps are far less quantified, and they also potentially have different sequestration timescales. So that may make this really important. But this was really a guess, kind of constrained by what you could find from the literature, but there are very few studies that have actually measured all these at the same time. One of the areas where we do have enough data to start putting actual numbers on all of this is the California current ecosystem. So this is a study where we put together the data from the CCE LTER program, and we quantified the fluxes out of the surface ocean, out of the and then into the deep ocean for sinking particles, active transport by vertical migrants, and subduction pathways. And what we see is that the gravitational pump is the most important, leaving the euphotic zone. It's maybe doing 60% of the export, and it's also going quite a bit deeper than, in particular, the subduction pump. The subduction pump was second most important, but really only got stuff down to like 100, 150 meters, so it didn't lead to much long-term sequestration. So in this graph, the orange numbers are the amount of carbon that's actually sequestered due to each of these pathways, and so we see that when we talk about sequestration, sinking is really the most important, but with active transport by the vertical migrants becoming second most important, because those organisms are respiring their carbon at usually depths on the order of between 300 and 600 meters. And now I want to kind of start talking about how we can understand each of these different pathways. So first, we've got the eddy subduction and mixed layer pumps, which are really the circulation-driven pumps. This is a figure from a study by Maureen Levy. This is a modeling paper. And models are really kind of the most tractable way at the moment to understand these pumps. And Levy et al. Split it up into, the eddy mixing, horizontal advection, vertical diffusion, and then entrainment and detrainment of the mixed layer. And with the model, you can start to put numbers on this, and you can see that there's a lot of spatial variability. And with models, you can start linking this to changes in the circulation that may be driven by increased stratification or other processes. But how do we actually look for this in the observations? So this is coming from a study that used biogeochemical Argo floats. And so basically what they're looking for is evidence of discrete subduction. So if you look on here, you can see areas where the apparent oxygen utilization drops, where there's increased chlorophyll, increased POC at depth. So that's suggesting that there was a subduction event that transported things to depth. And this is great because you can start to see discrete events and relate those discrete events to things like eddy kinetic energy. The problem is that turning this discrete event into an actual flux requires a lot of assumptions, which actually can be alleviated a bit if you accidentally leave it on sampling every day like we saw yesterday. But for the normal 10-day return, it's a little bit more difficult to turn that into a flux. Another way you can use the biogeochemical Argo floats is by basically using the fact that we know there's a nitrogen mass balance. The amount of nitrogen that comes up must balance all the amount of nitrogen that's exported downwards. So if you look at the vertical gradients of particulate nitrogen and the vertical gradients of nitrate, The ratio of those two, if you assume that the water column is just being mixed in one dimension, gives you the proportion of total export that's due to this subduction event. But of course, there's a lot of issues because the water column is not just being mixed straight up and down. Sort of a lot of assumptions that go into this as well. But interestingly, if you do this for the Southern Ocean, you get a very similar answer to what Lort got using the other approach. One approach gave 19%, another approach gave 23%. So that there's at least some similarities there. Maybe something makes sense. We've also got another approach that you can use, particularly in areas that have high seasonality, in particular with a deep mix layer in the winter. You can look at seasonal accumulation of standing stocks. So this is from some of Craig Carlson's work back in the 90s in the Sargasso Sea. And basically they looked at DOC increases throughout the phytoplankton growing season and then assumed that it all gets mixed down in the winter. And so this suggested that the DOC transport may actually equal sinking particle flux in the Sargasso Sea. So this pathway can be potentially important, but we don't have a lot of observational ways to really get a great handle on how this is varying. Next, I want to go over to active transport and diel and seasonal migrants. I love showing this paper or this figure from one of Daniele's paper because it just so clearly shows the daily signal. Some zooplankton stay up on the surface all the time. Others migrate down to depth during the day to run away from their predators. And if you want to estimate active transport, you can go and you can use acoustics, or more frequently, you tow a net up here during the day, you tow a net or up during night, then during the day. You look at the difference in biomass. And then you use the fact that there are pretty strong relationships between biomass, temperature, and organism size. And so you can estimate what the respiration is. And from that, you can put together figures like this that have mean migrant biomass on the x-axis, respiratory flux on the y-axis. And you can even, because a lot of these studies have been done at the same places that we're deploying sediment traps, you can compare respiratory flux to the total POC flux that's measured. And you can see that the respiratory flux is typically in the range of like 10 to 30% of the total flux. Although sometimes it does get to be about half of the flux. And because we know these relationships, we can start making some assumptions based on the ecology of the organisms. So we can take what we know about net primary productivity, and from satellite, we can also kind of estimate the fraction of large phytoplankton in the phytoplankton population. We know that the big zooplankton that vertically migrate are usually eating the large zooplankton, or sorry, the large phytoplankton. And so we can apply some different models. And so this is a figure from the Nowicki et al. That Tim talked about yesterday. And we can look, this figure shows the proportion of the migrant pump export divided by the total export. And if you look at this, first off, you see that across the globe, it seems it's generally less than 20% of the export. Second, another important thing is that it looks a lot like the fraction of large phytoplankton, which makes sense because the migrants are eating the large phytoplankton. And we can apply this figure from Bianchi et al. To estimate how deep they're actually going. So we actually know where they're exporting this stuff. So this starts to make sense. And, you know, they're eating the large phytoplankton. Large phytoplankton like not lots of nutrients. So maybe we can predict what's going to happen in the future. And we should expect ocean becomes more stratified, less nutrient flux, less large phytoplankton, less vertical migrators. So this active transport may go down in the future. But if we actually go to the places where this has been measured, if we look at the Bermuda Atlantic time series or the hot time series, zooplankton are actually going up. So I don't know what's going on. There's another really big uncertainty here, and that's the mortality rates at depth. Most of the estimates that I showed you are based on just saying, well, how much they breathe out at when they're at depth. And I mean, that's something we know. It's similar to us. Like, I can estimate how much we're all breathing out. I cannot estimate very well when everyone's going to die. And it's actually much more certain with zooplankton, because imagining when zooplankton are going to die is kind of more like imagining, well, when are we going to die if I release a few lions in here? And so it's much less uncertain. This is from one of my data simulating studies where we basically took all the data that we had available, taking all kinds of different rate measurements, used it to try and constrain the model. And with that, we constrained the fraction of active transport that's due to mortality. And it went from 0% up to about 98%. So really, it was just completely unconstrained. So how can we start trying to constrain that important term? Well, Robeson et al. Used that deep water time series that Colleen showed us yesterday, and basically took one approach of basically saying, hey, we've got estimates of the abundance of this siphonophore. It eats these vertical migrators. What's the capture radius of this siphonophore? So we can actually estimate what's the likelihood that a zooplankton gets eaten as it's running the gauntlet from the surface to the deep. Another approach is to couple some in situ measurements to a basic food web model and ask, well, what does the mesopelagic demand tell us what's going on? So this study showed that if you look at what's supplying the bacteria with their energy, most of the exports supplying their energy was coming from these passively sinking particles, with active transport not being super important. But if we look at the myctophids over here, the midwater fish, they're actually relying mostly on the active transport because they can feed directly on the vertical migrators. So that's another approach that we can use. And then this is from the exports program. They use compound-specific isotopic analysis of amino acids to basically look at what is the ultimate source of energy supporting the deep water organisms. And basically their results suggested that something like on the order of maybe 20-ish% of the midwater organisms' energy was coming from vertical migrators based on the isotopic analysis. And I'd also just like to remind everybody that this vertical migration stuff is important for more than just carbon export. This is from one of Amy Moss's studies. And basically the important thing to see here is this is the bacterial community in the deep ocean in their control experiments. This is the bacterial community where you allow copepods to pee. So you get much greater diversity when you allow these vertical migrators into your system because they're basically creating microscale patchiness and creating places where the specialists can thrive. So, and another reason that this is important is these vertical migrators are often stopped by the low oxygen zones. So they're often respiring right near the edges of OMZs. So Daniele showed that you actually increase the thickness of the OMZs because of the activities these vertical migrators. So knowing whether it's the subduction pump, the vertical migrators, or sinking particles is important for many different reasons. Now getting on to the gravitational pump. So this is a figure from Bissler et al. And it's a nice one. They set up on the y-axis, this is the EZ ratio. So this is the proportion of net primary production that gets exported. And here we have the transfer efficiency, the proportion of the exported material that makes it another 100 meters deeper in the ocean. And they told us some things like large cells, high export, small cells, low export. Export may vary across the lifetime of a bloom. Vertical migrators have high transfer efficiency. If there's not vertical migration, you have lower transfer efficiency. If there's a lot of bacterial respiration, you have low transfer efficiency. If there's not much bacterial respiration, you have high transfer efficiency. I want to remind you why sinking particles may be particularly important. This is from our California current study. This is where sinking particles fall on this plot. And remember, up here is high export efficiency. Down here is low transfer efficiency into the deep ocean. Here's where the active transport falls. It's high transfer efficiency, but not a large proportion of the export leaving the euphotic zone. Here's where subduction falls. It's very low transfer efficiency. So it's the sinking particles that are really getting things down deep into the ocean. We've also seen a figure like this. This is from Henson et al. This is showing the change in export flux in the CMIP6 models, just reminding everybody that there's a huge amount of uncertainty. There's a factor of about 3 uncertainty in what's going on right now. They tend to show similar trends, but that's really because they're all lacking the same processes. And so we can see estimates of how things may change in the future, but this should be considered pretty uncertain given the variability between the models right now and all the things that the models don't include. But I want to talk about some of the things that may be really important, some of the faces that we should put on the biological carbon pump, because these are the organisms that may be driving things. So this is from a study by Decima et al., where down in the Southern Ocean subtropical front, she conducted some experiments in waters, in very similar waters that either did or did not have salps. These big fun organisms that are basically gelatinous barrels that pump water through themselves and collect it in belly, and that's really all they do. They're just big ocean vacuum cleaners. But when you have salps, you actually increase the export flux by, on average, about a factor of 5. So huge change in sinking particle flux if you have these salps. If we move a little bit further in the Southern Ocean to the Western Antarctic Peninsula, they actually showed that their long-term time series of POC flux was strongly correlated with actually krill size. So it's when you have big krill, that's when you have a lot of export. So these particular organisms are really important. So maybe we need to know how those organisms are changing as a result of climate change. We now have the ability to sequence what's in our traps. So we can actually start looking at all the taxa that are in our traps. This is a really cool study that I like. We heard yesterday and from Henry a few days ago about the high flux events at Station M. If we look at those high flux events and look at the sequences in them, normally there's a lot of diversity in the communities on the sinking particles, but when there was a high flux event, it was all really dominated by one diatom, Thalassiocyra. So maybe Thalassiocyra is really important. Another study in a similar region by Beji Valencia compared the communities in shallow sediment traps to the mixed layer communities. And here, if we look at this, we see the one taxa that was consistently overrepresented in the traps was Thalassiocyra. So maybe it's this species important, but if we look, the other diatom species that was really abundant, another group of Bacillariophytes, was actually underrepresented in sinking particles. So maybe it's not just diatoms that matter, maybe it actually matters which diatom it is. And we can start looking at what other studies have found. Amaker et al. Found that Synechococcus is consistently overrepresented on sinking particles relative to Prochlorococcus. So this is something that's fairly consistent, is that if Prochlorococcus is dominating, you tend to have lower flux. Prochlorococcus tends to not be the dominant thing you find in sediment traps. Gutierrez et al. Found that radiolarians were dominating the export flux in their study. Farnell et al. Found that sinking particles were rich in multiple different types of diazotrophs, which kind of makes some sense because those diazotrophs can make their own nitrogen. Heterotrophs can also be important. Both et al. Found an important role for Syndiniales, which is a parasitic type of ciliate. And Fontanez et al. Found that pathogenic Vibrio taxa were potentially important. So maybe these pathogenic and parasitic groups are really important. But I do want to add one word of caution, and that comes from Beji Valencia's work. And it showed that actually a lot of the taxa that we find overrepresented in sediment traps are the taxa that are digestion resistant. So maybe there's a little bit of a preservational bias going on. And we need to be somewhat cautious when we interpret things. But still, we see these taxa that pop up again and again, and maybe important to export flux. So maybe we need to think about these individual taxa. This is a really interesting study coming out of the Bermuda Atlantic time series, where they've got their great time series. You can see the temperature increasing. You can see the ocean becoming more stratified. There's less nutrient flux. You see a shift towards more cyanobacteria dominating the community as the eukaryotes tend to decrease. So all of this would kind of tell us there's going to be less export flux. But they actually saw that although net primary productivity decreased, the export flux stayed high. And the reason the export flux stayed high is that when you got this switch to cyanobacteria dominating the community, those cyanobacteria had a high carbon to phosphorus ratio. So even though the flux of phosphorus into the euphotic zone was decreasing substantially, and the flux of phosphorus was decreasing substantially, the carbon flux stayed the same because the carbon to phosphorus ratio of the sinking material had decreased. And the carbon to phosphorus ratio of the sinking material had increased. And the reason it had increased is because of Prochlorococcus. So Prochlorococcus was the reason that export flux remained relatively high. So that thing I just told you about Prochlorococcus reliably leading to less export, that's also wrong. I don't know what's going on. And of course, the other really important thing to think about with the gravitational pump is flux attenuation. What's going on after the particles leave the euphotic zone? What determines how deep they make it into the ocean? This is one of the big uncertainties. We have way less measurements of this. This is a figure from some of Debbie Steinberg's work where she looked at the role of both bacteria and zooplankton as two groups that like to chew on sinking particles, although they do it with very different mechanisms. And basically, the takeaway from her study was that in one region in the North Pacific, they had roughly equal roles, whereas at the Hawaii Ocean Time Series site, the bacteria were substantially more important. but depending on where you are, which is most important can vary. And I also want to point out that different taxa of these groups can do things differently. So this is from one of our studies, again in the California Current, where we looked at the roles of different zooplankton. So one group of zooplankton that we looked at is the suspension feeders. The suspension feeders are the ones who are just going around, catching any particle that they come across and eating it. So they're feeding in a concentration dependent manner. And if you're feeding on a concentration dependent manner, then the amount of things you eat depends on, or your likelihood of eating something depends on its retention time in the depth zone that you live in. So this shows the percentage of flux consumed as you get to deeper depths in the water column as a function of, on the x-axis, the particle sinking speed. So what you see is the slowly sinking particles, and most particles are sinking very, very slowly, get almost entirely consumed really quickly. You have to get up to at least like a 10 meter per day sinking speed before you escape these suspension feeding zooplankton. So this is part of the reason that quickly sinking particles are so important. The things that we usually find in sediment traps are sinking probably at like 100 meters per day, because if they're sinking slower, they get eaten by these suspension feeding zooplankton. There are other types of zooplankton that have a very different feeding mode. We refer to them as flux feeders, and they're basically organisms that sit there like this, waiting for food to fall on them. So this is from Limacina helicina, so one species of pteropod. And it shows the cumulative percentage of flux intercepted. And for that, it doesn't matter the sinking speed, because they're basically an interceptor. They're just sitting there waiting, and they'll catch anything that's falling onto them. And so you see this one species can consume, before you get to 400 meters depth, anywhere between like 4 and 10% of the sinking particles. We also, in the same study, we looked at the phaidarians that Colleen was showing us yesterday, and the phaidarians basically have a similar impact. I can wrap up pretty quickly, I think. So, of course, the other side is bacteria. We've already shown this slide, so I'm not going to talk about it too much, but I think it's important to keep in mind that the bacteria are doing complex things ecologically. They're attaching and detaching from particles. They're growing and being eaten. And they're also producing exoenzymes that act on particular types of molecules. This is an unpublished figure from one of my grad students' work, in which she used ultra-high resolution mass spectrometry to look at the molecules inside sinking particles. And the percent island of stability is basically a measure of how stable molecularly particles are. And what she found is a really strong correlation between stability and overall flux. So we can think of this as kind of a molecular clock and the flux. And across regions, she basically found that it doesn't matter how deep you are, it matters how much flux there is. And this is really telling us something about the fact that diagenesis, the reworking of particles, starts in the euphotic zone, and the same processes are working in the euphotic zone and the deep ocean. And I'll just leave you with, again, this figure from Henson et al. I think it's good to go back to this paper every couple of months and remind yourselves of all the things we don't know and all the important processes that we've talked about that just aren't included in these models. And that's it for me. And I'll turn this over to Mara, who's smarter than me and hence hopefully knows something.
So next we have Mara Freilich, who's going to talk about hotspots of connectivity from the surface to the mesopelagic, from eddy fluxes to climate.
Great, thank you. And I'm honored to get to follow Mike and appreciate all of his insights here, which we'll be building on. Okay, so the fundamental question that I'll be talking about is what are the mesoscale and submesoscale mechanisms that are connecting biogeochemistry of the surface and deep ocean? And a lot of what I'm doing is inspired by satellite images like the one in the background that show really clear connections between biology and physics, where there are these small scale swirls in biology that suggest some coupling with the physics. And we have this surface view from satellites. The subsurface view is less well known and less well constrained. But what's critical is that the gradients in biogeochemistry subsurface are actually stronger than those that we see at submesoscale and mesoscale at the surface, typically. And so subsurface, we have the euphotic zone and a mesopelagic zone. We know that there's general increase in most nutrients, most dissolved nutrients. And then many of the organic quantities like particulate organic carbon or labile dissolved organic carbon or oxygen are highest at the surface and decrease subsurface with strong gradients, right, driven by light. And that's largely because these gradients in these biogeochemical properties are controlled by biology and air sea fluxes, right? And so in the surface ocean, we have photosynthesis, respiration, decomposition that recycles these nutrients and is what consumes them and causes these low nutrients or high particulate matter in the surface. The surface ocean and surface mixed layer is also where the ocean encounters the atmosphere, where we have these air-sea fluxes of biogeochemical quantities. It's also the zone where submesoscale and mesoscale processes are most important. And so when we're talking about connections between climate and atmospheric processes in the deep ocean, we need to understand the surface processes in the surface mixed layer. Could you start the clock for me? Thank you. I got some bonus time, I guess. And so what I'll be talking about today is really flux. And in that sense, thinking about flux meaning vertical motion across these gradients. And I tend to write this as W prime, N prime, right? Transport of anomalies in the vertical and anomalies in our tracer quantity. And there's a physical flux, and it's important to keep in mind that there is also a biological flux. These biological processes, again, are modifying these gradients. And so on the broad scale, we think that new production, as Stukel was just telling us, that new production and export production should be related to each other somehow, that our nutrient supply should relate to the amount of export we have. Observations have shown that we're so far not able to balance new production and export production, particularly these studies are from the subtropics, but I think this holds true across much of the ocean. And so that, I would say, is a key target in our understanding of some of these surface processes. And there's been increased attention to the idea that mesoscale and submesoscale processes may help us to close this budget. When I'm thinking about why does this matter, I'm thinking not just about biological carbon flux or flux that leads to sequestration and storage, but also the supply of carbon from the surface ocean is what is feeding some of these mesopelagic ecosystems. And so the amount of carbon that's fixed in the surface ocean where there's enough sunlight for photosynthesis and then that can be transmitted into the mesopelagic ocean determines how much productivity we'll have in the mesopelagic ocean and can set the stage for some of that transfer efficiency of carbon through this zone, but also just the ecological state of the ocean itself. And so the first key point that I want to get across is that eddy processes, these mesoscale and submesoscale processes, act on tracer gradients, and these tracer gradients are maintained by biological processes. And so there's inherently already a biophysical coupled dynamic here and result in a vertical tracer flux in the presence of vertical velocity. For the rest of the talk, I'm going to talk about characteristics of these eddy fluxes from both observations and models, just sort of what are we trying to understand. Then I'll talk mechanistically about some of these eddy flux processes, and finally conclude with some of the biological consequences. And so some observations. What observational evidence do we have that mesoscale and submesoscale processes matter for these connections between climate and the deep ocean? So one, on the left-hand side, I'm showing observations of biogeochemical variance. So this is showing oxygen or AOU variance on the horizontal axis with depth in high eddy kinetic energy regions in green and low eddy kinetic energy regions in purple, and just showing that that variance increases, particularly in the mid-depth when you're in a higher eddy kinetic energy region. On the right-hand side, I'm showing model studies, which are showing the extent of oxygen minimum zones as a function of model resolution, and just showing that there is significant sensitivity in oxygen minimum zone size. This is in the Arabian Sea, but this holds in other regions as well, to the model resolution, so whether you're resolving eddies or not. Like Stukel mentioned these inversions. So while we have a typical depth structure of high particulate organic carbon and high dissolved oxygen at the surface and lower carbon and lower oxygen subsurface, there are these inversions where we see these anomalous high AOU and high carbon subsurface. And that's been one of the kind of key points of evidence that there's a coupling between eddies and these biogeochemical vertical transport. So that's true in the surface mix layer. There's also been work dating back quite a while on what we call nepheloid layers that have seen transport of sediment out from deep slopes for the most part, and see inversions in carbon then in the deep ocean, so near the bottom boundary layer. In this talk, I'll mostly focus on the surface ocean, but I wanted to make sure I included this idea that these are processes that are happening in the deep ocean as well. I'm talking about boundary layers in general, and a lot of the physics that we understand from the surface mixed layer we can translate to bottom boundaries as well, although with some important differences. And so we have this 1D picture of inversions. One of the things that we were able to do then was to go out and sample at really high resolution. This is using towed profilers crossing a front. And so this is a 60-kilometer transect you see on the left-hand side with depth. And I've highlighted where we see high chlorophyll going along a sloping isopycnal surface, with the isopycnals here in the black lines, from the deep chlorophyll maximum into the interior. And so if you took a profile through that, you would see an inversion. When you look at it in this high resolution transect view, you can begin to get a better understanding of the process, and in particular that this is an isopycnal transport process. And so going back to this really nice paper by Nowicki asking kind of what is the integrated impact of what they call these mixing pumps, or these physically driven carbon export, find that there's quite a lot of regional variability in their importance as well. In some regions, there can be a really substantial fraction, up to 50% of the total export, particularly in boundary current areas or high eddy kinetic energy areas. But in other regions, they suggest that these processes are not important, not very important. And so we can build up this view where if you have a 1D profile, you see an inversion. But these eddy processes are really operating in 3 dimensions. And this is really critical when we think about climate change. So one of the ideas is if we have climate change, then we will have increased stratification of the surface ocean. If we have increased stratification of the surface ocean, particularly increased stratification of the surface mixed layer, that may inhibit exchange from the surface into the interior. These eddy processes, since they operate in 3D, they may be able to overcome some of that increased stratification and may be able to sustain transport from the surface ocean or the photic zone into the interior. And so as a quick summary of this, eddy fluxes increase variance of biogeochemical or other tracers near boundaries and occur in 3 dimensions and occur along density surfaces. So one of the key things here that I've been talking about is vertical velocity. And I'm not going to give a detailed lecture on physics of sub-mesoscale or what are the key vertical velocity processes. But I do want to talk about some of the scaling and why we might see some of the regional variability in the importance of these eddy subduction processes. Here's one way to look at what happened in the ocean. So if we are in 3D turbulence, the vertical velocity and the horizontal velocity have very similar spatial scales. As we go to mesoscale and sub-mesoscale, what we find is that the horizontal velocities are much stronger than vertical velocities, and that's true on the global scale as well. And some mesoscale is really in that boundary where we can have vertical velocity of about 100 meters per day. And so we're beginning to loosen some of the constraints of rotation. And I want to point out that that puts us in a different scaling on both raw speed number and the aspect ratio. And there's a zoo of different sub-mesoscale processes that operate, right? And I think a lot of really important work on what are the mechanisms at sub-mesoscale, and then how do these mechanisms at sub-mesoscale connect to particular biogeochemical impacts. I'm not going to go into detail on what each of these is, and I'd be happy to talk more about these and which matters. But what I want to do is kind of put this into a framework of saying what are these, if we look at their fundamental level, how do these relate to tracer transport and biogeochemistry? And so when we're thinking about tracers, one of the key things that distinguishes tracers from, say, energetics overall is that a long isopycnal transport is really key. And motion along sloping isopycnals matters, whereas if we're just concerned with stratification of the ocean or some of the physical state, then we're really just concerned with motion of the density surfaces themselves, and less so with motion of tracers along isopycnal surfaces. And when these isopycnal surfaces are sloping, then this energetically favorable along isopycnal motion has a vertical component to it, not just a lateral transport along flat density surfaces. And so let's look at that density framework in a little bit more detail. In a place like Bermuda, here is the depth and density in a 1 degree box near Bermuda from Argo profiles. And the photic zone there is about 100 meters. And so if a water parcel is going to move along a density surface to depth in this region, how deep can it get? And this is one way to approach this key question of how deep do these surface and boundary intensified processes, mesoscale and sub-mesoscale, transmit into the interior? So we take our line at 100 meters, we say we have to move along a density surface, so we have to move in the vertical. And that brings us down to about 400 meters in this particular location. If we take slightly denser water, maybe about the densest water that we find at 100 meters, then that gets us even deeper to about 550 meters in this region. And so 550 meters is solidly into what we define in most regions as the mesopelagic ocean. And in work that I've been doing with Lily Dove, we're defining this intermediate area between the photic zone where there's not enough light for photosynthesis. And so these biological processes focus more on respiration than photosynthesis. But where there is still easy access along density surfaces into the photic zone as a sort of biogeochemical transition zone between the photic zone and the mesopelagic. And that region is characterized by high biogeochemical variance as a result of this biophysical coupling. And so if we look at this in a little bit more mechanistic framework, we have this idea that we have a long isopycnal motion, we have restratification, and the restratification may differ if we're in the surface mix layer, where density surfaces are vertical, or if we're in the interior, where we're in a more mesoscale regime. And the sorts of parameterizations that we use are in most modeling is a mixed layer instability framework and mixed layer instability parameterization for the surface ocean, Gent-McWilliams bolus velocity for the interior restratification, and what we call a ready diffusion for our long isopycnal diffusion of tracers. We asked that question in models, though, of how do we get water parcels to cross that boundary? So to go from the mix layer into the interior, and does it fit within this sort of general diffusive framework? Does it fit within that general diffusive framework that this diagram would suggest? And so in this modeling study, what I'm showing here is density contours and 3 different model snapshots. And each of the colored dots is showing a water parcel that left the surface mix layer on the day that it left the surface mix layer and stayed down in the interior for a significant amount of time, in this case, about 10 days. And what you see is that it's not happening uniformly. Instead, it's happening in small scale features, so in some mesoscale features or features on scales of about 10 kilometers, but on mesoscale eddy, so on the areas of highest density gradient at the surface. And so this is suggesting to us that subduction, or this exchange from the surface ocean into the interior, is occurring intermittently along mesoscale fronts. We found that it occurs as these fronts intensify, so it's associated with frontogenesis, and that it is occurring in submesoscale features. One of the things that I think is important to keep in mind is that as this is happening, so as these water masses are going from the surface into the interior, what I found in this modeling study was that generated a sort of mixed layer source water mass at a rate of about 0.2 to 0.3 Sverdrups, and so much smaller than deep water formation rates, but it was contributing a fairly significant subduction flux. In other regions, there have been studies that say that these mesoscale and submesoscale processes can be quite important for water mass formation, in particular mode water formation, rather than deep water formation. And so I've been emphasizing that this along isopycnal vertical transport really matters. This is another modeling study I led where on the vertical axis here, I'm showing what is the proportion of the total vertical velocity that's along sloping isopycnal surfaces rather than just due to heaving up and down of isopycnal surfaces or to net restratification. And we find that that's sensitive to model resolution, where the diamonds are a low resolution model and the circles are higher resolution model, although both are high resolution compared to climate models. And we find that this scales with that aspect ratio that I was pointing out previously and the slope of the isopycnal surfaces themselves. So we can explain this. And when we combine this idea of the scaling of a long isopycnal vertical velocity and some of these ideas of biogeochemical variance at depth, I'm almost done. What we find is that we can construct a global estimate of the particulate organic carbon flux using observations. So using the global biogeochemical and traditional Argo floats. And so this is giving us an estimate of what is the particulate, this map is showing an estimate of what is the particulate organic carbon flux from these frontal processes alone. And we can compare it to sinking flux, and we find something that's actually quite similar to the Nowicki results in terms of the regional variability of the importance of this process. I made an assumption about geostrophic balance, and so I've not looked at the equatorial region here. And I think doing these sorts of kind of fundamental geophysical fluid dynamics scalings, applying them to observations can be one way to help integrate observations and global models and inform us about the ways that the models are sensitive to the choices we're making about the physics and the ways that the physics couple with the biology. And so here I'm showing a few different modeling studies at the top, looking at the connection between carbon and vertical iron flux, which is sensitive to both the model resolution and the parameterizations that they've chosen. On the bottom, how this along isopycnal diffusion affects the anthropogenic carbon uptake in global models, so the choice of both the magnitude and the spatial variability of those parameters, and then again thinking about why are these models so sensitive to the model resolution when we're asking about biogeochemical tracer fluxes. And so I think getting a better mechanistic understanding of that from studies like I showed can help to improve our estimates of which of these models should we be trusting and how can we improve these models to improve our estimates of the transport of biogeochemical tracers from the atmosphere into the deep ocean. And so with that, on the mechanistic perspective, we found that these coupled mesoscale, submesoscale dynamics are what's connecting the surface and interior. And I'll very briefly, so I won't talk about my results here, but just very briefly point out that this is something, like Mike Stukel was saying, that can affect the community structure as well, as we carry not just biogeochemical properties, but also total surface communities from the surface into the interior. And there are a number of reasons why that could be influential for the biogeochemistry of the mesopelagic. And so in summary for that, we find that vertical velocity is really a fundamental ecological factor that shapes mesopelagic ecosystems on subseasonal timescales. And before I wrap up, I would like to acknowledge Lillian Dove, a postdoc at Brown who's worked with me on this, and then Amla Mahadevan, my PhD advisor, who did a lot of this work with.
Okay, thanks so much, Mara. And Mike, are there any questions for our speakers? Go, Can.
Yeah, I mean, it's maybe a clarification question in general between physics and biogeochemical fields. When we talk about from the physical side, we talk about, I mean, as we discussed earlier today too, deep water formation sites and deep convection zones and all that stuff. Which biogeochemical fields are directly related to those deep water formation sites in their deep pathways, and which ones are not? I'm a bit confused because some of them seem to be related to those, some of them maybe not. Is there a clear distinction between the types of biogeochemical fields versus physical deep water formation sites?
Yeah, so that was actually a question that I wanted to ask yesterday as well. And what I what I talked about here really, I mean, I showed it. It's within the wind-driven gyres, and I was really focused on subtropical regions because that's where I've worked as well. And so one of the reasons when we were talking about the biological consequences that matters is most of the, say, POC flux attenuation is happening in those areas. That's where we have this mesopelagic biomass. But I think it's really important to keep in mind this sort of general principle that this covariance between biologically created gradients, which in the case of deep water formation could be both in the vertical and in the lateral, and the deep water formation matters. And so I think one thing, like if we have change it from a biogeochemical perspective, if there's changes in the timing of formation of deep water, that could affect what its biogeochemical content is when it is formed and when it leaves the euphotic zone, for example. And so I don't know, I haven't studied that, but I think sort of keeping that in mind is what I would say.
I don't know.
I have a question for Mike. I saw your last slide about the stability versus POC flux. It's really interesting. It's sort of consistent with, I think, Colin's work about the subtropical gyre where we have low flux but higher transfer efficiency. So I guess my question is, first of all, why? That's like chemical composition, I assume. And second, you showed bats, there's increasing carbon export. But what about the transfer efficiency? Like, will chlorocaucus sort of increase the transfer efficiency or the other way around?
So I guess there's two parts. The first part is about the molecular stability, and a lot of that, so I didn't get the time to go into it, but we were looking at the island of stability, which is a class of molecules found in all deep water DOM, and the assumption is that it's kind of the most stable molecules, and we found that that was very strongly correlated with export flux, and we'd expected that you would have more stable molecules deeper, less stable molecules shallow, and we did find that within a location. But if you compared the high flux upwelling region to an offshore region, at 450 meters beneath the upwelling area, the molecules were less stable, still juicier than right below the euphotic zone in an oligotrophic area. So that's really telling us that particles are getting acted on before they even start sinking. And when you really think about it, that makes sense. It's not like bacteria are not chewing on particles while they're in the euphotic zone. The ecology starts as soon as a particle is formed. And we, at least I as a biologist, make this distinction in my head of euphotic zone, shallow, below euphotic zone, deep, different processes happening. The biology doesn't care about that. The phytoplankton care about that. The bacteria, if there's food, they're going to start chewing. So that's kind of that part. Getting at the question of the transfer efficiency. So I do think that these questions are related. And I think that in part, there's an offsetting thing happening. In the areas, in those upwelling regions, you have juicier things, so the bacteria are probably cranking faster. They're chewing on things. I mean, it's like my son when he's eating Skittles as opposed to my son when he's eating Brussels sprouts. He's going at it when there's the candy. And that same thing is probably happening with the bacteria in upwelling regions. And the bacteria are probably chewing slower when they're feeding on the stuff that's coming out of subtropical gyre. But on the other hand, the particles beneath the upwelling area are sinking faster. So those two things offset. You tend to have smaller things sinking slower in the subtropical gyres. So slower bacterial activity, but slower sinking particles. And so those two things offset. And so I'm not sure which drives the transfer efficiency more, but it's really important if we're thinking about the ecology of the deep ocean, that we're transporting yummy molecules in beneath an upwelling zone. And we're transporting things that organisms really don't like to eat in the oligotrophic areas.
But the thing is, upwelling, you have like a larger particles. They're supposed to sink faster. So if I think about it naively, we should have a higher transfer efficiency. But I think in your case and in most cases, we sort of see a lower, like, right? Getting
and
deep.
I think that's, I mean, I'm not sure that we don't have enough estimates of transfer efficiency. And like in the Marseille et al., I think it's 2015 PNAS paper, which is kind of one of my favorites on that. They basically show that depending on the assumptions you made about how to extrapolate the transfer efficiency, you've got very different results because we're undersampled. And so how you parameterize it really matters. So I'm not sure whether transfer efficiency is higher in upwelling areas or offshore. I don't think we've fully constrained that yet. And I think that, yeah, you'd expect the fast sinking particles, higher transfer efficiency in the upwelling regions, but I think that's offset by the fact that you've got this yummy material that's sinking, so the bacteria eat it faster. So I think we have both things happening. And there's also higher abundance of zooplankton beneath the upwelling areas. So I think there are multiple confounding factors. And so once again, I don't know what's going on.
I guess I just maybe a yes and moment briefly, where the bacteria actually are very strongly depth structured, at least in the upper ocean. And so one of the things we found in our observations is we were able to sample on Lagrangian trajectories as they went from the surface ocean into the interior and sample the communities using sequencing. And so the phytoplankton are obviously strong differences between the surface and depth, but there are also really strong differences in the bacterial community composition. And we found that we had this detectable signal of bringing surface bacterial communities into the interior along these pathways, and those communities are primed for those high POC. And so if you had sinking flux through them, maybe they would remineralize differently. We also found a consistent change in the community composition. So we saw some of that changeover of communities as the phytoplankton are dying out, right? And so I think the bacterial story is important as well. And I think as Colleen was pointing out, in terms of who the bacteria are on these pathways.
Okay, great. We've got time for one more quick one.
So when we look at satellite pictures of chlorophyll or ocean biomass and NPP and so forth, we see rather large values at these frontal regions in the Southern Ocean that are, I don't know, south of 45 or something different. Can you explain this high productivity based on your collective work? And would you expect there to be also, yeah, I guess with the community structure, more diversity, less diversity, more grazed, less grazed. Yeah, because I can only see kind of the total phytoplankton being higher, and we expect it will be higher, bigger phytoplankton, larger phytoplankton there. But just in terms of maybe the physics, is this consistent with your work, I guess, at the smaller scales?
Okay, I can say briefly some things I know about that, which are that we do see changes in community composition across gradients consistently across the Southern Ocean. From a physical perspective, in these areas of deep water formation, those are also areas with relatively deep mix layer where you can mix nutrients up from depth.
Just the frontal structures corresponding to different temperature lines, right? 45 south, 50 south, going across the ACC.
Oh, so you're saying right at the
Not
front?
deep water, right at the front. I'm talking.
Yeah, yes. So then in that case, I think the general paradigm is that there's relatively strong vertical velocity at the front. And so you can bring up nutrients from depth. And I think in the Southern Ocean, that includes both nitrate, but also iron, and the concept that iron may be sourced from depth as well, and that's a significant frontal transport there. The figure that I had on the iron and carbon, that was from the Southern Ocean, and showing that if you both resolve and parameterize eddies, you can improve representation of carbon in the Southern Ocean.
And I'll go ahead and yes and that one. I personally think the vertical advection is probably the main driver of the increased phytoplankton biomass. But the other thing at those fronts is that they often separate, like the subtropical front actually separates nitrate limited waters from iron limited waters. So when you mix those waters, you have one water that has excess iron, one that has excess nitrate. So that can also stimulate additional production at the front, because now you have water that has both nitrate and iron. So you can get additional growth there. And you also asked about zooplankton and grazing, I think. And so there can be nonlinear impacts there because those fronts concentrate the phytoplankton. And so a lot of higher trophic levels will also congregate at the fronts. And so the actual impact on the export flux may be greater than the increase on phytoplankton biomass because you have so much higher zooplankton right at the front. So I haven't looked at this in the Southern Ocean front, but in one of the fronts in the California current, we did zooplankton net tows just going across the front, and right at the front, like you didn't have to measure anything. You just looked at the bottle and you're like, oh, the front has way higher zooplankton biomass because they congregate there. And then you've increased the zooplankton and you've increased the phytoplankton, seen counter rates go way up.
And this is regardless of the type of front? I mean, if you're in upwelling, downwelling regime, or?
I don't want to say that.
Yeah, okay.
I think it depends on the depth of the front and what biogeochemical gradients it's crossing. And so at front, you will have both upwelling and downwelling. And there's important interactions with wind as well that are going to control those dynamics. But yeah, I think it's, again, keeping in mind how the physics are relating to the biogeochemical gradients.
We are 5 minutes into our coffee break, everyone.
That's important.
Okay, let's thank Mike and Mara again for 2 excellent talks.